Hello, everyone, and welcome to today's live broadcast, Simultaneous Single-Cell Visualization of RNA and Protein, presented by Sara Becker-Catania, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by eBioscience Affymetrix. eBioscience Affymetrix technologies enable multiplex and simultaneous analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of benchtop research into clinical and routine use for human health and wellness. Affymetrix, part of Thermo Fisher Scientific, provides leadership and support, partnering with customers in pharmaceutical, diagnostic, and biotechnology companies and leading academic, government, and nonprofit research institutes in their quest to use biology for a better world. To learn more, visit www.ebioscience.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Sara Becker-Catania. Dr. Becker-Catania is an Associate Director in R&D at eBioscience, where she leads a team dedicated to the development of immunology-focused reagents for flow cytometry and microscopy applications. She received a PhD in experimental pathology at the University of California, Los Angeles, studying the genotype and phenotype correlations of a rare neurodegenerative disorder, ataxia telangiectasia. Dr. Becker-Catania did her postdoctoral training as a Giannini Foundation Fellow at the UCLA Neuropsychiatric Institute Mental Retardation Research Center, studying the selected specification of neural stem cells along neural and glial lineages. Following her postdoc training, she held a joint appointment as a research assistant professor at the University of Illinois Chicago and the Heinz VA Hospital, and was a recipient of an Illinois Regenerative Medicine Institute stem cell grant for her work with neural stem cells in models of multiple sclerosis. Please join me in welcoming Dr. Sara Becker-Catania. I will now turn the presentation over to her.
Thank you, Judy and LabRoots, for the opportunity to speak today about the development of a new assay that allows us to look at visualizing RNA and protein in single cells. Cellular heterogeneity is present in any biological sample, and whether it be at the level of gene expression, protein, or differences in post-translational modifications, most of our understanding of gene and protein expression has been from methods that really look at entire populations and give an average result. While this is really useful and it's been a foundation for science leading up to this point the last few years, scientific methodology has really advanced, and it's advanced our ability as researchers to understand the role and the contribution of single cells. Over the last 5 years, we've seen an increase in publications and embracing the single-cell approach to all types of models in biology. And it really started out in 2013 in Nature Methods as they published the method of the year, which was single-cell sequencing. And as we can see over publications in different areas of biology, a marked increase in the number of publications that look at single-cell biology in an effort to really understand the role of the rare cell. These are sometimes masked in studies where we've looked at population averages. So this was a real limitation. In our R&D department here in product development at eBioscience, we already utilize a lot of techniques that look at single-cell biology. We use flow cytometry, we use in situ hybridization, immunocytochemistry, and immunohistochemistry on a daily basis. But a problem that we came across was really looking at understanding the role of an mRNA or an RNA species in the same cell as protein. And so when we looked and using RNA-ISH, and we currently have a assay that does this, this is the ViewRNA Cell Assay, we can easily detect messenger RNA in the figure on the left. And we can detect protein using antibody-based methods like immunocytochemistry as shown in the center image. But when we tried to combine these two assays, we did not get the results on the right, which would have been a merge of the two methodologies. And the reason for that is the two protocols that are used for immunocytochemistry and in situ hybridization are very incompatible. So it either results in a destruction of the RNA or a destruction of the antigen that the antibody recognizes. So the challenge was really finding a method, a protocol and reagents that would allow us to do both in the same cell, in the same cell cultured in a dish. So what I'm going to show you in the next slides was the development of that assay and a protocol and the solutions to really look at message and protein in the same cell. And this assay is what we call the ViewRNA Cell Plus Assay. So it's a novel assay. It uses multiplex fluorescence to detect protein and RNA in single cells. And this group of images really shows the breadth of the assay and the different types of cells that it's been used to look at different proteins and RNA. Everything from cervical adenocarcinoma cells to virally infected epithelial cells shown in the top right, as well as models of circulating tumor cells, neurons and glia, and epithelial cells. So all of the different types of cells that a researcher might encounter in a typical look into their model of interest. The applications of the assay are far-reaching. It allows us to analyze sample heterogeneity. We're interested in looking at mixed populations, whether it be looking at one rare cell within a mixed population, or even understanding the differences between what we think is a homogeneous population, for example, a cell line. And I'll show some data later in the presentation that really will make you think twice about what we accept as a cell line and accepting that we think that it's homogeneous when perhaps it's not really that homogeneous. It allows you to correlate RNA and protein kinetics within the same cell. We've done some studies looking at non-coding RNAs, including microRNA and long non-coding RNA in cellular subsets. We've analyzed messenger RNA expression levels when there isn't an antibody available, which commonly happens. And it allows us to track and visualize viral RNA. And there is the potential for high throughput compound screening if the assay is adapted to a platform that's used in those types of applications for imaging. If we look at the workflow of the assay, this is an overview. So on the left-hand side, sample preparation and antibody staining is the first step in the protocol. So samples can either be grown in 96-well plates or in chamber slides or on cover slips in 24-well plates, or they can be suspension cells that have been cytospun onto slides. Then at this point, the sample is fixed and permeabilized in a single step. Antibody staining is done, and sometimes secondary staining of the antibody if that's the choice that the researcher chooses. And I'll go into more detail on this in slides coming. And then the next step is really detecting the RNA of interest. So we do this using specific probe sets designed for the RNA of interest, and they can either be in 3 different types. And the type of the probe set really determines how it will be detected for microscopy. So a type 1 probe set will use an amplifier and a preamplifier and label probe that is a type 1 label probe, and this is detected in the Alexaflor 546 channel on the microscope. Whereas a type 4 corresponds to an Alexa 488 detection, and a type 6 corresponds to an Alexaflor 647 fluorophore. So these are sequential hybridizations where we can look at multiple RNAs within the same cell as well as antibodies. And then the readout, the detection portion of the assay, is done using either a standard epifluorescent microscope or a high-content imager. In looking at more depth at the experimental workflow, it's really broken down into 2 days. In this case, step 1, on day 1, this portion of the protocol here looks at detection of the protein of interest, where it's using antibody-mediated, basically immunocytochemistry, to detect the antibody. Then a fixation step is done, and then the assay proceeds into hybridization to look at the target RNA, a hold and wash buffer overnight, and then on day 2, this is an amplification of the detection of the RNA signal. So the total assay time on day 1 is 6 to 8 hours, and on day 2 is 4 to 5 hours, and this doesn't include imaging time. And the hands-on time is 2 to 3 hours on day 1 or 1 to 2 hours on day 2. In looking at more depth at the protein detection strategy, there are a couple different ways to think about performing the assay, and there's a lot of flexibility built into the assay. So one can use a direct protocol, and in direct I mean that the antibody that you're choosing to use to detect your antigen of interest or your protein of interest in the assay can be directly conjugated to a fluorophore. This is a nice way to go because the protocol is faster. You don't have to use a secondary antibody or a secondary antibody with a streptavidin, as in the 2-step and 3-step protocols shown below. The beauty of the direct protocol is really the ease of use. If you want to look at multiple antigens and they're both, for instance, a mouse IgG1 isotype, it becomes very difficult to do a 2-step protocol where you're using an anti-mouse secondary because you'll have cross-reaction between the 2 primary antibodies. So if you're using a direct protocol, it's very easy to use one antibody that's labeled with an Alexa 488, for example, and another one that's labeled with an Alexa 647. But it doesn't mean that that's always the best way that you should do it or the only way that you can do it. And there are a lot of antigens that may be expressed at very low levels, which require a 2 or a 3-step protocol to really amplify the signal and to really be able to detect the antigen of interest. In which case a 2-step or a 3-step protocol is advantageous simply because it allows you this amplification of that signal of antigen. And either method is applicable in this assay. There's no limitation to that. It's just whichever is the right approach for your antigen of interest. And this really depends on the level of expression of that antigen. So in choosing the antibody, there are some criteria that are very important. So the first step is really to identify which antigen or antigens are of interest to you and to identify candidate antibodies that will work in the assay. So the criteria for the antibodies is that they typically perform well in an immunocytochemistry assay. So these aren't necessarily antibodies that work for immunohistochemistry or formalin-fixed paraffin-embedded tissue. Those may be different antibodies. But these are typically antibodies that work for cells that are grown and fixed in a dish. The antibodies that tend to perform very well in this assay are antibodies that perform well under formaldehyde fixation and permeabilization conditions. Antibodies that are used following acetone fixation or methanol fixation don't tend to work very well with this protocol that's been designed. And that's just one limitation of the protocol. Most of the antibodies that are available for immunocytochemistry tend to fit into the formaldehyde fixation and permeabilization group. So there is a lot of flexibility and a lot of different specificities that can be performed with this assay. So you can either use a purified or a fluorophore conjugated, as I spoke about in the last slide, and that really depends on whether or not you have a fluorophore conjugated antibody to the specificity that you're interested in, and also whether you think that that antigen is expressed at low levels and may need the amplified signal that you would get from using a secondary antibody or a secondary biotinated antibody with, for example, a streptavidin conjugated to a fluorophore. The next step is really to test the performance of the antibody on your cells using the ViewRNA Cell Plus fixed and permeabilization buffer set. So we sell just the buffer set alone, and this is specifically for the purpose of testing your antibodies to make sure they can perform well in the assay. There's limited value in doing the whole assay if the antibodies that you're using don't perform well. This also allows you to titer your primary and secondary antibodies, which we recommend anytime you're running an assay where you're looking at maybe a different cell type and you haven't run that particular antibody before. You should always titer the primary and secondary antibodies and decide if you're going to use fluorophore conjugated antibodies. The next step is then to perform the whole assay with the RNA detection. And for validated antibodies, we have a list of clones and fluorophore conjugated antibodies that have already been validated in this assay, and this can be found on our website at the address below. So as we look past now the detection of protein and we look at the detection of RNA, the RNA detection is done using a branched DNA amplification. And this is really the basis for the ViewRNA cell assay that we currently sell that just allows you to look at RNA. But in this particular assay, what we have is we have detection of RNA using a target probe mix. So basically these are probes that are designed specifically to the target RNA of interest. And it requires a paired set of these to bind to the target RNA, and then with each sequential addition, first preamplifier is added, which binds to the extender region of the target probe and forms the trunk of this structure that will be formed that will allow you to detect the RNA. The next step was the addition of the amplifier, and the amplifier are really the branches that bind to this preamplifier. And the last step is the addition of the label probe. And the label probe binds to the amplifier. And from each set of target probes, 2 of these probes, you can have the addition of 400 label probes onto this tree structure, which really allows you the amplification to see the RNA within the cell. So the target probe sets are designed to be highly specific and sensitive. So first, you would need to determine your RNA of interest. You need to have an accession number for that, and then decide which probe type you would like for it to be. So probe type, as I said before, refers to the type of detection channel that you would like to see your RNA in. A type 1 is similar to a TRITC or an Alexa 546. Type 4 is similar to an Alexa 488, and a type 6 is an Alexa 647. So then the Affymetrix Biometrics information team identifies regions within the RNA that you're interested in that would give the highest specificity over a range of about 1,000 nucleotides, and designs a probe set composed of about 20 oligo pairs that are highly specific for your RNA of interest. So the probes are manufactured. Some of them are already commercially available. These are the same probes that are used for the ViewCell assay. And if it's required that they be synthesized, it takes less than a week to get the probes. So the next thing I want to do is show you some of the specificity data from these probes. So if we look at the specificity of the assay, we know that it requires the binding of both of those probes, a probe pair set, to really form the tree structure. So in this case, what we've done is we've looked at an mRNA GAPDH, which is typically a housekeeping gene. It's used a lot for that purpose. And what we've done here is we have taken the left probe and the right probe, and we've hybridized them alone. And you can see in the 2 images on the left and in the center that there is no corresponding signal of RNA So the RNA signal here would be white, and you can see the only thing you see are the nuclei of the cells, which are stained blue for DAPI. And then on the right-hand side, the right-hand image where you see the white punctate dots, each dot corresponds to an individual mRNA, GAPDH mRNA. And the image on the right is done under standard conditions where we've used paired probes, the left and the right probe together, to yield the signal. And then we followed with the addition of the preamplifier, the amplifier, and the label probe to really build up that tree structure, which really allows us to detect the single RNA species. So these images were taken all at the same exposure time. So they're matched exposures so we can really truly evaluate the amount of signal between each of these different conditions. In this case, the second example is specificity. We've gone ahead and on the left-hand side, we omitted adding the probe. So this is a no probe control. And you can see that when you omit that probe, you do not have nonspecific binding of the preamplifier, amplifier, or label probe. The tree cannot form without and bind to the cells without the addition of the probe. This image in the center is the DAP-B control. And DAP-B is just an E. coli gene, and so it should not be expressed. These are human HeLa cells, and so there should not be any expression of DAP-B mRNA in the cells. And we can see that there's no signal present. The only thing we see are the nuclei of the cells. And then on the right-hand image, we see the GAPD positive control, and this is just GAPDH staining here in the cells. This is showing individual mRNA, and this is the expected signal on the right-hand side. And again, these images were all taken at the same exposure time and shows really that there is no nonspecific binding of the preamplifier, amplifier, or label probe components of the assay without actual binding of that initial probe pair. In this case, what we're doing here is we're looking at a comparison of the ViewRNA Cell Assay, which is the assay that's currently sold by Affymetrix, which allows you to look at RNA, and then a comparison to results of the ViewRNA Cell Plus Assay, which allows you to look at RNA and protein. So these cells are HeLa cells. The probe set is designed to ERB2, which is the gene that codes for the mRNA that codes for the HER2 protein, which is important in breast cancer. And what we're looking at in the two images are probe sets designed to either exons 2 through 9 in red, and this is a type 1 Alexa Flora 546 probe set, and a second probe set, which is a 488 probe set of type 4, and this is designed to exons 10 through 20. And the reason we did this was to look at paired red and green dots. So seemingly a paired red and green dot would indicate a single mRNA, where we have tree structures formed on the 5' end of the mRNA and also on the 3' end of the RNA. And it's very difficult to look at these images overlapped to see the two signals. But if we look, here I'll try to point to this. For example, here, oh goodness. If we look up here, we can see two dots up here corresponding to two dots up here. And these actually are paired dots where we see the single mRNA species. And what we did was we went ahead and quantitated the amount of type 1 and type 4 dots that we see between the two assays. And we counted 6 cells. If we look at the average number of dots per cell, and the reason why we can quantitate it this way is because the ERB2 message is expressed at very low levels in these cells. So it allows us to see individual dots and count them. If we look at the amount of expression, the average number of dots per cell, in the View standard assay, type 1, which is the pink box, and we look at the red bar next to it, the red bar is the View Plus assay, we see that the average number of type 1 and type 4 dots isn't significantly different between the two assays. If we look at type 4 in green, the light green is the ViewCell assay and the darker green is the View Plus assay. It's not a significant difference between the number of dots that are detected between the two assays, showing that there's a nice correlation between results of the two assays. This is an actual comparison looking at a housekeeping gene and basically two housekeeping genes, beta-actin and PPIB. And both of these are, the beta-actin type 1 is in red and the PPIB type 6 is in white. And I have an inset box in each image showing the PPIB expression singly, just imaging that channel alone so that you can see it, because sometimes it's hard to detect the white on top of the red signal. So if we look at the Cell Plus assay, which is shown in the top left image, we can see detection of beta-actin mRNA in red and PPIB in white. Those are the individual dots. And then the nuclei are counterstained with DAPI in blue. And the top right image is the ViewRNA Cell assay. And we see similar expression of and detection of beta-actin type 1. And PPIB, we can see in the inset box, is expressed in white. So we have a little bit more sensitive detection of PPIB type 6 in white with the View Cell Plus assay. And then if we were to go ahead and look at the actual image of the View Cell assay merged with the protein, in this case we looked at expression of alpha-tubulin using an antibody directly conjugated to Alexa 488. And that's shown in the image on the bottom left, showing expression of tubulin in green and beta-actin in red and PPIB in white, along with DAPI counterstained nuclei. If we compare the expression of protein between the View Plus assay and just standard immunocytochemistry, we see there's a very nice correlation between the two. The top row of images is the ViewRNA Cell Plus assay, looking at three different specificities, Golgi protein, COX4, and Ki67. The bottom three images, the row of images, is standard immunocytochemistry, just using antibody, and in this case, all of these were done, these are paired reagents, so using an antibody to Golgi eFluor 660, which is similar to an Alexa 647, and this is imaged in green here, and we see very nice expression between the ViewRNA Cell Plus assay and standard immunocytochemistry. These are the left top and the left bottom images, and we're looking at the green signal, which is the Golgi protein, which localizes to the Golgi apparatus, and you can see the nice localization in the cells. The center images are COX4, Alexa 488, is the antibody that was used, and this is a mitochondrial protein, and this is expressed throughout the cell cytoplasm. And we can see in the immunocytochemistry assay, the signal is just a little bit brighter. So sometimes in the Cell Plus assay, the combined assay, you do get a little diminishment of signal of particular proteins. And this is highly protein specific, and it varies. Most of the time, we don't really detect a hit in the signal, and this is still quite usable to detect COX4 protein. But I point it out because there can be some differences when you're combining two assays, one for looking at protein and one for looking at RNA, that you may not get exactly the same performance as you would with immunocytochemistry, where you're just looking at protein levels. The last two images on the right are looking at Ki67. So Ki67 is a protein that's involved and is expressed in actively cycling cells, and it's expressed in the nucleus, and it's shown in white here. And it often appears as this punctate white staining in the nucleus. And we can see that there's very nice concordance of signal between the two assays. So that this is using a directly conjugated antibody to an Alexa eFluor 660, which is similar to an Alexa 647 fluor. And that's shown in white. Next, we looked at the ability of the assay to assess a heterogeneous population of cells that was in suspension. So these were mouse splenocytes, and they were treated as a cytospin preparation. So the cells were isolated and they were spun down onto slides, and the assay was performed on slides. So what we were looking at was a population, a mixed population, and we're looking for the expression of CD4. So CD4 is expressed on lymphocytes, and we can see in the left-hand image expression of CD4 mRNA in white, and these are the punctate dots indicating single copies of CD4 mRNA. And in the center image, we see cells that are staining pink very lightly. So this is surface expression of CD4. Since lymphocytes have very limited cytoplasm, the expression of CD4 or any surface marker in a lymphocyte, when you look at it by cytospin, often looks like it's expressed right on the nucleus, and it's because the cytoplasm is so scant. And in the merged image on the right, we see that the cells, only the cells expressing CD4 mRNA look red or pink, and indicating a nice correlation between the CD4 mRNA and the CD4 protein. And you see that cells that are other types of cells, maybe they're CD8 positive, maybe they're macrophage, they don't express the CD4 protein in this mixed population. We wanted to look at some different models that researchers typically use for their studies. So one of the ones that we picked was a myoblast differentiation model. And this is using a mouse C2C12 cell. So undifferentiated, these cells, if they're put into differentiation conditions, will give rise to muscle cells. So they're often used to study myogenesis or metabolism or muscle biology. And they're actively kept under high serum conditions, which let them proliferate in culture. And then when the experiment is done, they're plated onto cover slips, and the serum is removed and they're put into a low serum condition, and this causes the C2C12 cells to differentiate and form myotubules, and they do this by upregulating myogenin. So when we look at the assay performance in this population, here we're looking at differentiation of myoblasts over 5 days. So the top row of images shows day 0, day 3, and day 5. And this is looking at proliferation. So to look at proliferation, we used an antibody to Ki67, which marks proliferating cells. And this is a directly conjugated antibody to E4660, and it's shown here in pink. It's pink because the signal is actually red, and it's co-localizing with the cell nucleus, which is stained with DAPI, which is blue. So when there's co-localization between the two, it appears pink in the image. So we can see that the number of pink cells is decreasing as we move from day 0 to day 5, as the cells really start to differentiate. They stop dividing and start differentiating. We also see that the amount of myogenin, which is shown as white dots, increases in cells as we move from day 0 to day 5, indicating that these cells are upregulating myogenin and starting to differentiate. If we look at the differentiation markers on the bottom row of images from day 0 to day 5, we see that the amount of myogenin protein, which is shown in green, and this is using an antibody to myosin Lexophor 488, is actually increasing. And we can see the number of green cells has increased at day 5 compared to day 0. And also we can see that the number of green cells with myosin protein also have myogenin mRNA, indicating that these cells are actively differentiating. The red in this image is just beta-actin mRNA, just used as a housekeeping control in our study here. In this slide, what we've done is we've looked at myogenin protein and mRNA in the same group of cells, and also with beta-actin. So the protein is myogenin, and it's conjugated to Lexophor 488, and that's shown in green. And you can see the number of green cells in day 0 is a fairly low percentage. And then as we look at day 3 and day 5, we see the number of cells that actually express the protein and the myogenin mRNA, which are the white dots, is actually increasing. And we can even see the start of the expression and the formation of myotubules, which is the classic phenotypic thing that these cells do as they start to differentiate. So this is shown, let me show you a myotubule here. So myotubules have multiple nuclei spread along what appears as a long continuous cytoplasm where these cells have fused together to form these myotubules. And I think that this really highlights the heterogeneity of the culture. So we see that not all the cells are actively differentiating. Some are still at this point dividing, as we saw in the previous slide, and yet some have started this program for differentiation. So I think in thinking about the way we use the cultured cells and cells that we think are more homogeneous, we really need to understand and accept that perhaps those cells are not as homogeneous as what we think. And I think this is a really nice illustration of that. Okay. So everything that we've looked at up to this point for the assay excuse me, has really been looking at mRNA. But we know that mRNA are really a small proportion of the total RNA that are present in a cell. So the majority of RNA in a cell are actually non-coding. And this has become more and more important in the last few years as we've seen the emergence of the role of microRNA and of long non-coding RNA. And so this was an area that we were really interested in the lab, and we really wanted to put this assay to the test to see if we could detect microRNA. We also did some work looking at long non-coding RNA. I'm not going to present that today, but we were able to successfully use the assay to also look at non-coding RNA, the long non-coding. So as many of you know, microRNA are very small. They're approximately 20 to 24 nucleotides in length. They don't code, but they actually end up regulating gene expression by directing target mRNAs for degradation or translational repression. So all of the microRNA probes that we use are all type 1. This is detected on the TRITC channel of the microscope, or Cy3. And it's because there's a very nice signal-to-noise ratio when we use this fluorophore. The probes hybridize to either mature microRNA or the pre or pri microRNA sequences, which are the unprocessed forms of the microRNA. So if we look at some of the different microRNAs that we looked at, we were really interested in understanding the microRNAs that are involved in breast cancer. We were currently making some antibodies looking at proteins that are important in breast cancer. And we wanted to pair that with some of the work that we were doing, and we really wanted to understand some of the microRNAs that had been identified as having a role in either causing breast cancer or some role in response to breast cancer therapies. So these were just a few of the microRNAs that we looked at, and we presented some of this in a poster earlier this year at the AACR meeting, where we really looked at a whole panel of microRNAs and their expression in different breast cancer cells. And this is just an example of some of the data that we found. So in this case, on the top row, we looked at Ki67 expression. So this is really looking at cycling cells, and it localizes to the nucleus, and it's shown in pink. And then we looked at microRNA 107, and this is shown as the signal in white, and these are the white punctate dots that we see. So the antibody and the microRNA is on the left, and then in the center we have our housekeeping gene just used as a control, and the merged image on the right really showing the three, the Ki67, the beta-actin, and the microRNA 107. And then in the bottom row of images, what we have is detection of LET7 microRNA. And we see this microRNA is actually expressed at higher levels than miR-107, shown above. And these were just two of our initial experiments to really see if we could detect microRNA using this protocol that we were using for the assay. In this slide, This is data taken from the poster that was presented at AACR in April this year. And this was really a look at a panel of microRNAs in different cell lines. The top row of images shows an example of just very steady state levels of a microRNA, in this case microRNA 145. And this was similar to other microRNAs that we looked at, like miR34A, miR143, where the levels in these different breast cancer cell types were actually very steady state. We didn't see anything remarkably different between them. So these are all breast cancer cell lines. The SKBR3, BT474, MCF7, and the MDA-MB175 all fit into a category that's more a luminal type. So these display a estrogen receptor signature, and they resemble luminal types of tumors. The MDA-MB231 cell line is a more basal breast cancer line, which displays more of a BRCA1 signature and resembles basal-like tumors with mesenchymal and stem cell characteristics, actually. So the center panel of the center row of images actually is looking at expression of anti-survivin, so survivin protein in green. And this has a very interesting localization pattern. So survivin is actually an inhibitor of apoptosis. It inhibits caspase activation, and it's associated with tumor reoccurrence and poor patient survival. But the protein itself actually localizes to the mitotic spindle and the cleavage furrow, which I think you can see in some of these images here. For example, here in these two cells, and we see that the microRNA that we looked at here, miR107 in white, is actually differentially expressed at higher levels in the BT474 line and also the MDA-MB175 line. So these were similar to results that we had seen in the literature from Riaz et al., where they had looked at array results, so expression array studies looking at this microRNA in breast cancer. And we were able to confirm that study using this assay. The bottom row of images shows a look at miR221, and this is shown in white as well, and an antibody to Ki67, which is shown in pink. And again, this is using a directly conjugated antibody in the assay. And we see higher expression in, well, actually low expression in the BT474 and the MDA-MB175 lines. It's very difficult to detect here. It's actually fewer than 5 copies per cell. And then, so overall, what we see as a whole is a low expression in luminal cell types, which are the 4 cell lines on the left. And then on the right-hand side, the MDA-MB231, we see a much higher expression of more than 20 copies per cell. And this really correlated well with the microarray and real-time PCR data that we saw for miR221 in the literature. So overall, the results from this assay are very consistent with the published microarray and real-time PCR data. As I mentioned before, we were developing some antibodies specifically to look at markers for breast cancer. And one of the things that we were working on was an antibody to HER2. So ERB2 is the mRNA that codes for the HER2 protein, and as many of you know, HER2 is very important in breast cancer therapy, and understanding HER2 status is very important for developing a therapeutic approach for any patient with breast cancer. So one of the things we did in this study was we took breast cancer cell lines that we knew had established levels of HER2. In this case, the SKBR3 and the BT474 lines, which are both classified as like a 2+ HER2 line, and then the MCF7 line, which is HER2 negative. And we went ahead and we looked for expression of ERB2 mRNA, HER2 protein in the center row, and the merged image is shown in the bottom. So the ERB2 mRNA is shown in white, and we can see that there's very consistent expression of the ERB-B2 mRNA in the 2 cell lines that are reported to express HER2 protein. And we do not see any expression in the MCF7 cell line. It got a little more interesting when we looked at HER2 protein, because what we found was that even though ERB-B2 mRNA is expressed in virtually all of the cells shown that we looked at of the 2 cell lines, what we saw with HER2 protein was that it was a lot more heterogeneous. We saw expression that localized to the membrane and the cytoplasm, and that was expected. There's a lot of times in these cancer lines you'll see a mislocalization of HER2 protein expression. And we saw protein expression, throughout the cells, but then there were some cells that had very little protein expression but had quite a bit of mRNA expression, which was rather unexpected. So when we looked at the merge image on the bottom, you can see that there are some cells where there is message shown in white, but there's not as much HER2 protein shown in red. And in this case, I think what we're seeing here is really a difference in the cells that are in the culture. So it's a representation of what is normally occurring biologically, and it may be that the cells are turning over the HER2 protein more quickly, or maybe the ERB-B2 message is just starting to come up and protein has not come up yet. Or in this case, it may also indicate, it may be assay dependent, indicating that the amount of amplification that we get using a probe for ERB-B2 is about 8,000-fold amplification. So it's a much more sensitive look versus using a protein, an antibody to a protein. In this case, what we did was we used a directly conjugated antibody. So we're not getting any amplification of that process. One antibody is binding to a protein, and so there's no amplification of using a secondary antibody, which would bind perhaps multiple primary antibodies. So that may also contribute to the results that we see here. And as I mentioned, for the MCF7 line, which is negative for HER2, we do not see any expression of the message or protein. We did a similar look for progesterone receptor, where we had a cell line that expressed progesterone receptor and one that did not. And we see that progesterone receptor mRNA is expressed in the BT474 line, and we see nice correlation with expression of progesterone receptor protein shown in pink, localizing to the nucleus. And it's a nuclear receptor, so this is exactly what we would expect to see. And in the MDA-MB175 line, we see that we do not see any message or protein. We also looked at estrogen receptor alpha. So in this case, we used 2 lines, one that had low expression of ER and one that has higher expression of ER, the MCF7. And we can see estrogen receptor mRNA present in all of the cells of the BT474, but the levels are extremely low, less than 10 copies per cell. And in the MCF7 line, it's much higher. The MCF7 line, we're able to easily detect estrogen receptor alpha protein, and this is using an amplification step. So this is using a purified antibody followed by an anti-mouse IgG1 secondary with its fluorophore conjugated. And we see nice expression in the MCF7 line of the ER alpha protein in pink. Interestingly, in the BT474 line, we aren't seeing that expression of ER protein, which may indicate that when the levels of mRNA are this low, we don't get actual production of ER protein. Or it may indicate that the antibody is simply not sensitive enough to pick up and amplify and show the signal of protein that's there. So when we looked at assay performance for RNA expression, what we found was, in this case, this is a photomicrograph showing ERB-B2 signal in white. These are the white dots, GAPTH type 4 is pink, and this is just a housekeeping gene, and Ki67 in green, which marks dividing cells. And then we went ahead and we counted 15 cells per well, and this was run in triplicate in a 96 well plate format. And if we looked at the corrected total cell fluorescence, so this is done by simply marking a perimeter around the cells and quantitating the amount of signal within that perimeter, and then taking an adjacent area where there are no cells and subtracting out any background signal coming from the slide itself to give us this CTCF, the corrected total cell fluorescence number. We can see that the average CTCF across wells is very similar. And the CV values that we actually see, the variance, is actually coming from the heterogeneity of the culture itself, showing that the number of copies of ERB2 and GAPDH actually varies from cell to cell. So to really make this kind of data more significant, one would need to analyze a lot of cells to actually approach statistical significance. And it just is important to point out because a lot of the models that researchers use are cultured cells and cell lines. And this is a typical cell line HeLa here where we see this variation in the number of copies from cell to cell. If we look at assay performance for protein expression shown in green here, this is looking at alpha-tubulin expression and again doing a corrected total cell fluorescence for alpha-tubulin across wells where we've counted multiple cells. We see that statistically there is not a statistical difference between the average CTCF between wells, and this is looking at groups of cells. So the assay is actually, performing quite well across different wells when done in replicate. Now I want to show you a little bit of data from some of our early access sites. And this is really exciting. This is looking at expression and infectivity, and this is looking at Zika virus actually. So these are Vero cells that have been infected with Zika virus and stained for tubulin. And we can see in the top row of images, these are uninfected cells. And I'll draw your attention to the image on the top right, where we're looking at Zika virus expression, and it should be in red. And these cells are not infected, so there's no expression of Zika virus. In the bottom row of images, these are looking at Vero cells that have been infected with Zika virus. And I draw your attention to the right-hand image on the bottom, where we see red punctate dots, and this is actually the Zika virus. And the merged image is shown on the bottom left, showing tubulin expression, which really shows the structure of the cells and the cytoplasm, along with those, the detection of the Zika virus in red. And this data is courtesy of the Brass lab, Abraham Brass at UMass Medical School. The next set of data is really looking at a model for circulating tumor cells. So these were, this is data from RareCyte. These are SKBR3, which is a breast adenocarcinoma cells that are spiked into a mixed blood cell population. And in the top row of images, what we see on the left-hand side is the merged image, and this is showing cytokeratin or epithelial cell adhesion molecule expression, which really marks cancer cells, and we see that those cancer cells are yellow on the surface. And then the image to the right of that, the RNA plus DAPI, this is looking at expression of GAPDH in green and ERB2, so mRNA in red. So this is HER2 message in red. And see, we see red and green dots expressed here in these tumor cells and not expressed in the neighboring cells, which are just stained with DAPI. And so the bottom row of images is simply another view within the same experiment of cells that also express the cytokeratin and the EpCAM on the surface, as well as the RNA and the DAPI, which really show these cells as cancer cells compared to the other cells here in the culture. So this is a really look at a rare population of cells within this mass of blood cells in the sample. The next set of data I'm going to show you is from Suzanne Tomdik, and this is from her work looking at dissociated rat hippocampal neurons and glia. And in this case, she was really interested in looking at neurons that are dividing and differentiating along an interneuron path versus a different type of neuron. And in interneurons express parvalbumin mRNA, and that's shown in the cell in the very center where she's used an antibody to microtubule-associated protein 2, which is specific for neurons, and that's shown in red. And then the parvalbumin mRNA is shown in blue, and you see blue punctate dots around that cytoplasm shown here. And then she's also used an antibody to GFAP, glial fibrillary acidic protein, which marks glial cells. So this is a mixed culture of glia and neurons where she's used 2 different antibodies and 1 RNA to really highlight the type of neurons that are differentiating in her culture. In this data, this is the same model. This is looking at 13 days in culture. And this is CAM kinase 2A, which again is an interneuron marker shown in red. And the exciting thing about this culture is, and this experiment, where she can really look at the amount or the populations of neurons that are differentiating in this culture. So in the bottom left corner, I draw your attention to these 2 cells here, where there's expression of red punctate dots, which indicate that this neuron is differentiating towards an interneuron phenotype. And the cell on the right of the arrowhead, where there are blue punctate dots, and this really indicates that that is along a more general neural lineage. So we see the specific interneuron forming as well as another neuron. And again, in green on this image are astrocytes, or cells that express the GFAP protein. So this is really exciting to be able to look at the differentiation, the different populations of neurons, and be able to differentiate them simply by looking at expression of this particular mRNA. So during the course of our validation efforts of the assay, we did a lot of work looking at different probes. We've used over 47 mRNA, more than 18 microRNA, 2 long non-coding RNA, and 2 viral RNA probes to really look at the performance of the assay. And I can say that all of those probes worked successfully. We didn't have any that didn't perform well. We've looked across multiple cell lines, and I think from the first slide that I showed you at the beginning, where we looked across different cell types, we've really looked at a whole smattering of both primary cells and cell lines to look at performance of the assay, as well as some of the more rare subtypes of cells, and even cells like PBMC, where we've actually cytospun the cells down onto slides. For antibodies, we've currently looked at over 110, 64 different clones of multiple formats. By formats, I mean conjugated to different fluorophores. And we've looked at structural transcription factors, a whole host of different specificities and localization within the cell. Within the antibodies that prefer formaldehyde fixation permeabilization, we have an 88% success rate in this assay. So overall, the performance highlights of the assay are you can use 2 RNA and 1 protein, or you can look at 2 protein and 1 RNA, or 3 RNAs. You can see single copy sensitivity at single cell resolution. You can have probes designed for any RNA target. You can use purified, biotinylated, or fluorophore conjugated antibodies. Any of them work within the assay with just minimal changes to the protocol itself. We get robust results in adherent or suspension cells across multiple platforms, so either cover slip or 96-well plates or chamber slides. And really it serves as a companion assay to typical fish assays or immunocytochemistry assays. For more information for the assay, you can look on our website or contact our tech support at either the number listed here or the email listed below. And online we have our user manual for the assay and protocol, a white paper detailing the validation efforts of the assay, and then a brochure on the right telling about some of the applications that the assay can be used for. And with that, I will conclude and can take questions now. Thank you.
Thank you, Dr. Becker-Catania, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Becker-Catania, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many questions as we have time for today. Let's get started. Our first question is, what is the detection sensitivity of cellular RNA in terms of the length of RNA and expression/copy number of RNA?
Currently, we've used the assay to detect RNA species that are as small as 20 to 24 nucleotides. The example I showed in the data was the microRNA. And we've also detected many mRNA where there's a region of about 1,000 nucleotides, which is used for designing multiple probes along that region. So we have a lot of flexibility in the type of species of RNA that we're looking at within the assay.
Can we apply ViewRNA Plus to FFPE sections, for example, of cardiac cells?
For right now, the assay has just been validated on cultured cells that have been fixed onto a platform, either cover slips or slides, or we can use cultured cells. Dissociated splenocytes have been used, PBMC that have been cytospun onto slides. It hasn't been validated for performance in formalin-fixed paraffin-embedded tissue at this point.
Is there any way to adapt this so that analysis of stained cells can be done by flow cytometry?
We currently sell an assay for analysis where the readout is flow cytometry, and this is called the PrimeFlow assay. And it's a similar assay to this, except that the readout is by flow cytometry, not microscopy.
All right, we'll just pause for a moment to see if some more questions are coming in. And just give it another moment here. You know, while we're waiting for questions, I would like to once again thank Dr. Becker-Catania for her presentation. Do you have any final comments? Oh, before you do that, another question for you. What is the cost to have a probe made?
Probes can be made. They can, I'll have to get back to you on the cost, actually.
Okay. The next is, what do you feel is the best normalization control for relative mRNA level assessment?
Typically, we use a housekeeping gene. We use beta-actin, GAPDH. Sometimes the levels of beta-actin can actually differ in cells, especially during the course of an experiment. We can see levels that actually fluctuate. So a lot of times we'll just pick a relevant housekeeping gene and use that. We have a list of housekeeping genes that have been validated for different cell lines, which is available on our website.
Okay, and again, as we wait for additional questions to come in, I just want to let our audience know that any questions we do not have time for today will be answered after the presentation. So while we're waiting for more questions to come in, I would like to, as I said, once again thank Dr. Becker-Catania for her presentation. Do you have any final comments?
I just wanted to thank you for the time today, and I'll be happy to answer questions, and they can, of course, be submitted. We'll follow up with the answers for those. Thank you.
Great. Well, thank you once again. I would also like to thank our sponsor, Affymetrix, for making today's educational webcast possible. Before I go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 17th, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.